So we're going to start talking about enzymes, but before I talk about enzymes, I really want to get into, again, that idea of moving energy around. I do think of ATP as a cellular battery. You can recharge a cellular battery. That would be like building the ATP. How many of you guys use rechargeable batteries at home? Yeah. I think in the end, they're way cheaper than having to keep buying new batteries. All right. They explode. It's not like a galaxy. You take these batteries out of the wall, you plug them into your remote control, your remote control starts working. You're using some of that energy. That energy is then decreasing in it. You can then, if you wanted to, move the battery from the remote, and you guys know you've done this at some point, to something else, because, man, you need that other battery. So you can keep moving these batteries from one point to another, but each time you move the battery, you're losing a little bit of energy. Until in the end, you're out of energy. So you have to recharge it. Energy can move around a system. And truthfully, studying the way energy moves through a biological system, a whole system, is ecology. I know a lot of you guys remember studying ecology when you were in middle school and you learned about biomes and the tundra and how things work like that? It's way less interesting at higher levels. It's all about studying the way electrons move. Okay, so I want to talk about, I guess for the remainder of the lecture, enzymes. Enzymes are proteins, which means what are they made of? They're made by ribosomes. What are they made of? Amino acids. You did. Well done. You win. Enzymes, like we said earlier, are catalysts. A catalyst is something that speeds up a reaction without itself being altered or consumed in the process. As you can tell, I was up late trying to put the slide together because that is not a biological catalyst. Google Images did not serve me well this time. What that is a catalytic converter on a car. But then I was like, I should remove this and really spend another half hour trying to find a picture of a catalyst. And I was like, no, this kind of works. Do you guys know what a catalytic converter does? Yeah, just magic. Yeah, it goes out and then the car doesn't charge the battery. Because you see arrows. So it breaks down pollutants. That's one of the things it does. Exhaust goes in, carbon dioxide. So you've got hydrogen, carbon monoxide, and nitrogen oxide go in. Carbon dioxide and nitrogen go out. And that's where you get your greenhouse gas of carbon dioxide. But truthfully, it's a lot better than carbon monoxide. That could kill you. Right. This is a good trade-off.
But what about when you're... Oh, wait, never mind.
So what ends up happening is these molecules are broken down at a higher rate into those molecules in the catalytic converter than outside. But on a macro scale, let's not even talk about catalytic converters. Let's just talk about mechanics and car problems, because we've all had that. A catalyst is something that speeds up reaction without itself being altered or consumed. How many of you guys have gone driving and that little check engine soon light pops up? Yeah, pretty much all of us, because we all have really crappy cars. You like your car? You've got...
I just got a new car.
Oh, is it like brand new?
It's 17.
7? That's really new.
Yeah, I know.
Did you get a check engine light on that?
It went away though. It fixed itself.
That's the question. What do you guys do when you get a check engine soon? Guys, what do you do when you get a check engine soon light up?
You freak out.
First thing you do, you freak out. You
And then you ignore it.
That's right. That's what we all do. Every single one of us will look at that thing and say, if I hit a pothole just right, it'll fix itself. Right? There is hope beyond hope that could occur. Or you assume the light itself is broken. Because it's an expensive fix, isn't it? So you drive along 24/7, this light stays on, nothing gets fixed. Sometimes it might because you tighten your gas cap. But the idea is, so you tighten your gas cap. So there you go. So this light stays on until you take it to a mechanic or you fix the problem on your own. If you didn't tighten the gas cap, if you didn't take it to the mechanic, we're pretty sure it'd stay on in perpetuity. Now, how could, is it possible it would fix itself? Yes, it is possible. It is in the realm of possibility that you could be, your car could sit in a field and the raindrop could hit it just right and there's a vibration or something and the whole car gets fixed. That is absolutely possible. It's possible. It could take 1 billion years, but it is possible. The thing is, you can make it happen faster. How do you fix your car fastest? You get it fixed. You take it to the mechanic. What does the mechanic do? They open it up. They run a diagnostic. It's going to cost you money. Don't get me wrong. But they can make this car work. In the process, does it happen faster than it would have happened on its own? Yeah. Is the mechanic then altered or consumed in the process? That is, do they lose like a hand to your car? Not if they're a good mechanic. They're not consumed by it. It's not like your car eats the mechanic in order to get better. So the mechanic acts like a catalyst. They speed up the reaction. And then the cool part is they don't just fix your car and they're done. They can go on to the next car and the next car. So if you have one mechanic in a group, that one mechanic can fix multiple cars, making all these people's lives easier, happening much faster. A catalyst, like the mechanic, will speed up a reaction without themselves being altered or consumed. And this is where I like to say there's an intersection of politics and science. Because you've probably heard the term catalyst outside of this class. Lots of organizations say they are a catalyst for change. In fact, I think John Tyler says that. We're like, we are a catalyst for change in the community. What people think catalyst means in modern parlance is they speed up change. But what they forget about is the second part. I went to a VCCS conference, Virginia Community College System conference, and one of the high muckety-muck vice chancellor, arch-chancellor, whatever they are, the people who are in charge, they're giving this speech, and they were talking about how the community college system is a catalyst for change in the region, and how we are all catalysts. And so I'm the jerk that just stands up, and I'm like, I have a question. And at first they weren't going to call on me, so I started jumping and waving my hands around until they're like, oh, did you have something to say? And I was like, yes. Do you know what catalyst means? And they said, yes, it means we change things. Like, that's not all a catalyst means. A catalyst means we change, a catalyst will change something without itself being altered or consumed. So if we, at the college, are saying we are catalysts for change, what does that mean?
They're not changing, but they're changing other things.
Right. So that's like me saying, I'm a catalyst for change. I'm changing you, but I'm myself not going to change at all. That's not a catalyst. That's not a catalyst at all. It could be. Well, then I was thinking, you guys have had professors at some point in your life, I assume, who sort of represent this whole, I'm going to change you, but I'm not going to change. They have like those like yellowing notes that they read from. You've been in those classes, right? Yeah. So maybe we are. That is the way it works. The truth is, though, in a college setting, my interactions with you guys change me just as much as my interactions with you change you. I get you to learn things, but I'm learning all sorts of stuff, like what works and what doesn't, and what I have to do to help you on an individual level succeed. So I don't think we can say colleges and professors are catalysts for change, because we are changing. So there we go. Politics. So enzymes are catalysts. That means that enzymes are going to speed up a reaction without themselves being altered or consumed in the process. They increase the speed at which reactions occur by many, many times. At this point, I want to discuss the parts of an enzyme, because Reagan left, and it's going to be easy to talk about when she's not here. So, there's the enzyme itself. The enzyme has distinct structures on it. It's got a shape. The shape of the enzyme is called its conformation. You need to know that the shape's called a conformation. It's kind of easy to remember because it's got the word form in it. So the form is the conformation. It's enzyme shape, shape of the enzyme, whatever you want to say. That's the shape of this particular one. They have lots of different shapes because each of them has a distinct form, giving them each a distinct function. Now, with this enzyme, you have a place where something can bind. That place where something can bind is called the active site. And then the thing that binds to the active site is called a substrate. So the enzyme has a specific shape, a conformation. Part of its conformation is an active site, and that active site can bind to a solute in the solution called a substrate. And it fits pretty darn well. Oftentimes what it'll do, like here we've got the enzyme, it's got an active site. Sucrose, a disaccharide, can bind to the active site. And that sucrose is split or broken apart into fructose and glucose. So the substrate is released, and you go back. You'll notice the enzyme itself doesn't change. It speeds up the reaction without itself being altered or destroyed. Say that again? We talked about receptors when we talked about the membrane. In the peroxisome, we have, what happens in the peroxisome? Do you guys remember? Speeds up things, breaks things down. What does it break down? Yeah, because it's on the slide. Breaks down hydrogen peroxide. It breaks it down, 10 quadrillion times faster than it would normally. So one little bit of enzyme, little bit of this enzyme called catalase, breaks down hydrogen peroxide 10 quadrillion times faster. That number is almost unimaginably huge, which means that we don't have the effects of that free radical because it breaks down faster than it's being made. This catalase, this sort of enzyme shape here, what is catalase? It's a protein that breaks down hydrogen peroxide. It literally grabs hydrogen peroxide and rips it apart. Well, that's exactly what we have in our wounds. That's why so you take hydrogen peroxide, you put it in your wound, that thing goes along and rips the hydrogen peroxide apart. Yeah, so we are totally fine. Our wounds can handle hydrogen peroxide, no issue. It's bacteria that can't handle them because they lack this. Yep. Yeah, they wipe out things that don't have catalase. Here's why we need enzymes. When two atoms or molecules bond, their electron clouds repel each other. So the deal is, when I told you guys about covalent bonding, it should have made absolutely no sense. For some of you guys, it actually did make absolutely no sense. But the idea is you have one atom and another atom. You bring them together. And what I told you was, when you bring these two electron clouds together, they share electrons, right? You guys are all like, yeah, that totally makes sense. That makes no sense whatsoever. Because if you have a negatively charged electron cloud and a negatively charged electron cloud, and you bring them together, what should happen? They should repel each other. But they don't. We're just like, but they bind covalently. The reason you need energy for reactions is because you need to overcome that repulsive force. If you were to take two magnets charged the same way and bring them together, they push each other apart, right? Can you get those magnets to squish together? Yeah, because you're strong enough to push them. Does that make sense? You add that energy to bring them together. And when you do that, then they stick. You have you were adding activation energy. Here, you're adding activation energy. The energy you need to overcome the repulsive force of those electron clouds. Oh, every single semester I do this. I meant to bring eggs. This would have been so cool if I had eggs. I'll go into what I'll do. Okay, so imagine, if you will, I had 2 eggs. This would have been brilliant. I would have moved this chair so you could see too. Okay. So I take 2 eggs, and I put them on this desk. And they're not hard-boiled or any tricks like that. They're just straight up eggs out of the refrigerator. And I gently roll them toward each other. What will happen? They'll roll toward each other, they'll hit each other, and then bounce off. Just like if they had electron clouds. They have that repulsive force. They're going to bounce off of each other. How would I get them to stay together? That's right. You take them and you go squish. And then I would have been like, there would have been a splash zone for it. It would have been great. But the eggs then, once you get them together, do they come apart? No. They stay together. You added that energy, and that forces them to stay together. You need to add that activation energy to get these 2 atoms to stay together, to overcome the force of those electron clouds. Once you overcome that force, they stay stuck. You have a covalent bond. So the point of enzymes is to overcome that repulsive force. You have to add it. What enzymes do is they lower the amount of energy you need to overcome that bond. They lower the amount of activation energy you need. They make it easier. So enzymes decrease the activation energy necessary for a reaction to occur. Which is what this slide says. So here we've got ATP and glucose. The ATP and the glucose are going to bind to a substrate, to an enzyme at the active site. And then they're going to be brought together. A phosphate is transferred to the glucose, and ADP, adenosine diphosphate, is released. So this speeds up this reaction. And the way this one speeds up the reaction is it literally will bring these 2 molecules together and then start straining the bond between the phosphate at the same time squishing it to the glucose so that it makes the phosphate switch from one molecule to the next. You're just transferring a functional group. Pardon? No, it's cool. What? Any? Oh, the one that says, why do we need enzymes? No problem. Slide 26. So enzymes are going to be proteins that are made from amino acids that have that very, very specific shape, and that shape is called its conformation. This P shape here is its conformation, its size, its shape. The conformation determines exactly what that enzyme will bind to. It's very, very specific. They're not general. So on the enzyme, you have a place called the active site. The active site is where you're going to bind to the substrate, the thing that you're interacting with. Again, the form determines the function. So the form of that active site determines what substrate it can bind to. Now, that specificity of the form connecting to the specific enzyme is called the locking key hypothesis. Enzymes are going to typically bind to one type of substrate. So the specific enzyme will bind to a very specific substrate, usually. And by controlling how much enzyme is produced and how much enzyme is removed from a system, you can control how efficiently the different metabolic pathways in your body work. So this is, again, lock and key. The substrates are very specific to the enzyme. Once a substrate binds to an enzyme, you have what's known as induced fit. The enzyme changes shape a little bit. Now it's going to release. You're not going to permanently change the shape. But can I use this cup real quick? Okay. So the idea is, generally, I need this sort of C shape to fit on that cup, right? If I was to come on and put a C shape here, though, it's not going to be able to pick it up. What needs to happen is it needs to curl around a little bit more, tighten up a little. So in order to lift it, you have the C shape binds, and then it tightens, and then you can lift. So that's called induced fit. You're changing the shape. And the reason this happens is because, remember, what's this protein made of? amino acids, and each amino acid has specific chemical properties associated with it. So those amino acids are going to interact with these substrates, causing them to shift around a little. You might have new disulfide bridges form. You may have new hydrogen bonds form, a new hydrophobic effect, or a new van der Waals forces. So it changes the shape of the enzyme just a little bit, causing it to wrap a little bit tighter around the substrate. All right, so that's what enzymes are. Environmental factors will play a huge effect in how well these enzymes work, how efficient they are. And there are 4 big factors that affect enzyme function, 3 of which we're going to cover in lab. Enzyme concentration, the presence of inhibitor, temperature, and pH. So concentration, inhibitors, temperature, and pH are 4 factors that you're going to need to absolutely know. And you know there's a list here. It's going to be on the exam. I'm just saying, that's an easy question. So first off, I'm going to go through each of these. Enzyme concentration. If you have one enzyme in a solution, it speeds up the reaction, right? What happens if you have 2 enzymes?
Double speeds it.
That's right. It makes it go even faster.
Oh, I see.
No, that's exactly right. That's what I was getting at. So if you have a single enzyme in solution, it speeds up the reaction. If you have more than one, if you increase the amount of them, increase the concentration, suddenly the reaction goes even faster. It breaks things down much more quickly. Does that make sense? On the other side, you might have inhibitors. Inhibitors slow down reactions. You have 2 broad types of inhibitors: competitive inhibitors and non-competitive inhibitors. A competitive inhibitor is a substrate that's in the solution that will bind to the active site. So if where did I just put that marker? There it is. Here's our normal substrate for our enzyme. If I was to have another inhibitor, it's got the same shape. If it was to bind there, it would stop the substrate from binding. You guys have heard of formaldehyde? Yeah. Formaldehyde prevents enzymes from working, which stops organisms from breaking down. So a competitive inhibitor competes with the substrate for the active site. So if you have a lot of competitive inhibitors present, the reaction rate decreases. A non-competitive inhibitor does not compete for the active site. It's non-competitive. It binds to what's known as the allosteric site, a different site. This is outside of the active site, and it causes a change in the shape of the enzyme. So if you had an allosteric site over here on the enzyme, and something comes in and binds there, when it binds, it will change the shape of the active site so it can no longer fit to the substrate.
So the substrate still doesn't bind.
Substrate still doesn't bind. Net result, no binding of the substrate, which stops the reaction. So the more inhibitors you have, the lower the reaction rate. Competitive inhibitors compete for the active site, stopping substrates from binding. Non-competitive inhibitors bind outside of the active site, but they still stop substrates from binding. So the more inhibitors you have, the lower the reaction rate. You have a whole bunch of other stuff going on in the environment, though. You might have pH. What's pH a measure of? The amount of hydrogen ions present. And you might have temperature. Do you guys know what temperature is a measure of? The measure of movement, the measure of kinetic energy. So you might have these 2 conditions, pH and temperature, that will impact the reaction rates dramatically. First of all, temperature. Oh, by the way, sorry, last slide. There's this word called optimal. Optimal means the best. There are like the best possible conditions. So enzymes work best in the environment in which they've evolved. What is your body temperature? Roughly. Fahrenheit, I don't care. 98.6. 96.1 if you're running cold, that's cool. People have different temperatures, surprisingly enough. My daughter, yeah, it's my daughter. She runs hot. Now, she knows she runs hot. That means that her temperature is always about 99.5. Right. And her nurse at school has never taken her temperature at a normal level. So whenever she wants to come home from school, she goes to the nurse. And the nurse takes their temperature and like, oh, she's running a fever. You have to come pick her up. Yeah. Like, stop it. You're giving in. She's playing you. They're like, I'm sorry. Rules are rules. She's running a fever. Nah. For her, or for Reagan, if your temperature runs at 99.5, your enzymes are evolved to work best at that temperature. Some people, 98.1. Some people, 97.4. Yeah, 96. Some people run low. Your enzymes are evolved to work absolutely ideally at those conditions. Here, we've got this is a graph of, this actually looks like human body temperature here. 37 degrees Celsius is human body temperature. Starting off really, really low, you'll notice there are low reaction rates. These molecules cannot connect with each other. As you increase the kinetic energy, the molecules begin to interact. They begin to cause reactions. That steadily builds until you reach what's known as the optimum temperature. The reaction rates are the fastest they will possibly be. Once you get beyond that optimum, the rates drop dramatically because you start to lose the shape of the enzyme, the conformation of the enzyme. So at low temperatures, you have low kinetic energy. The molecules and the enzymes aren't meeting. As you increase kinetic energy, you start to have the enzymes meeting the substrates, and you have reactions occur. Faster and faster until you reach that optimum temperature, at which point you get a sharp decline in the reaction rate. At very, very high temperatures, the bonds holding these proteins together break down. What are 5 forces that hold proteins together? Covalent bonds. Van der Waals forces.
Yes.
I figured that's what you're saying. What? Ionic bonds. Covalent bonds. We got that. Hydrogen bonds. And the hydrophobic effect was the 5th one. At high temperatures, those bonds start to fall apart. So we go from having a 3-dimensionally folded up protein into what's called a denatured protein, a protein without those bonds. It lost the shape. That denaturation cannot be fixed without what are termed chaperone proteins, and that doesn't happen often. So you go from a protein that has this very specific active site. That active site has a certain shape to it. As you increase temperature, the bonds break, and you lose the shape of the active site. So you don't have reactions occurring. Egg. Have you guys fried an egg before? Yeah. Okay, you fry an egg. You crack open an egg. What does the egg look like when you crack it open? It's sort of like clearish, right? And it's got a yolk in it, right? You put it on a frying pan. You increase the temperatures. The egg white begins to denature, and it starts to solidify and turn white. Can you unfry an egg? No, because you can't renature proteins. You lost the 3-dimensional shape. Another condition will denature enzymes, and that's pH. pH, again, is the measure of hydrogen ions. So if you have too many hydrogen ions, those are going to end up pushing against the bonds in the protein until the bonds break down and you denature. If you have too few, you have too many hydroxide ions that will break the bonds in that protein. You have to have just a very, very stable amount. How do you stabilize the number of hydrogen ions in solution? buffers. I always think, do you guys remember playing Red Rover, Red Rover? Yes. So with Red Rover, what happens is you form a chain, like an amino acid chain. You're going to have some people running at that chain. They might represent the hydrogen ions. Now, if you had one person running at you, usually you can hold it together. But can you imagine having, like, I don't know, thousands of people charging this chain of people playing Red Rover, Red Rover? They're going to let go and break. Yeah, because you don't want that. So that's denaturation. That, well, I wanted to, but it looks we're running late. So those are the 4 big things that will impact enzyme reaction rates. Concentration of enzymes. The more enzymes you have, the faster the reaction rate. The concentration of inhibitors. The more inhibitors you have, the lower the reaction rate. The temperature. An optimal temperature will have the highest reaction rate, while beyond the optimal, reaction rates drop off dramatically. And then the pH, again, optimal pH and beyond optimal pH, reaction rates drop off. One thing I do want you to understand is that at high temperatures, you cannot refix an enzyme. Reaction rates are low, and they will stay low forever unless you add more enzymes. At a low temperature, you didn't destroy the enzyme. They're just not moving fast, which means that if you heated it up, the reaction would begin again. That's going to come into play in your lab. And then finally, a quick slide on cofactors and coenzymes. You've probably heard of cofactors and coenzymes in commercials. Basically, what you have with a coenzyme is that coenzyme is a piece of a puzzle. You have an apoenzyme, part of an enzyme, that will bind to a coenzyme, something that you're going to eat, take into your system, or you build, and that will allow you to then fully bind to a substrate efficiently. So coenzymes are going to speed up reaction rates because substrates don't bind to enzymes as well without them.